Thank you everyone for joining this webinar. As the host mentioned, in the first talk, we will have a discussion about the VHH-based bispecific antibody discovery with a novel single B cell approach. VHH, also known as a single domain antibody, has become a popular modality for therapeutic antibody in recent years. It is much smaller than traditional full-length IgG and its Fab or single-chain Fv fragment, giving it superior tissue accessibility and flexibility in bispecific or multi-specific antibody engineering. Moreover, its unique feature of inner disulfide bond enables better developability and manufacturability. So far, single domain antibody is widely used in bispecific or multi-specific antibody and CAR T cell therapy for tumor targeting. In addition to the drug targeting, recently, single-domain antibody is also applied in the in vivo gene delivery for cell and gene therapy by conjugation to mRNA or fused with lentivirus. By the end of 2023, there are 4 launched products adopted VHH as the core function domains, followed by more than 140 active pipelines in the clinical and preclinical stage. Most of these pipelines are for oncology and autoimmune diseases. As of today, immunized phage display library is still the most well-established and popular approach to generate VHH antibody leads. Anybody recovered from immunized library usually comes with higher affinity, but the panning process will almost certainly lead to significant loss of diversity. In addition, you may also experience challenges in immunization when there is a immune tolerance issues in which the target shares high homology between the human and the camelid counterpart. Under such circumstance, naive library can be a better and quicker option because it is totally independent of immunization. It enables a jump start with direct phage panning and screening, results in a much shorter timeline and better candidate diversities, along with the sacrifice of antibody affinity at roughly 100-fold lower. Nowadays, we have developed a novel single B-cell based approach to combine these desirable features in one platform. It still relies on camelid immunization, but takes less time comparing to the immunized library. The antibody affinity is around the nanomolar to picomolar range with the best diversity. The success of single B-cell screening relies on 3 pillars. The first part is the isolation of antigen-specific B-cell. Instead of dumping all PBMC into the library, we take advantage of antigen-conjugated beads to enrich for antigen-specific memory B-cell, followed by in vitro activation of a memory B-cell to plasma B-cell to facilitate the second step, the supernatant-based identification of a positive B-cell conducted on the chip of a Beacon single B-cell screening platform. It can run multiple runs of antigen binding assays with recombinant protein or cell line within hours and avoid the majority of a diversity loss during the screening stage. Those selected positive B cells will be exported for further characterization by a high throughput expression and profiling system called ProSpeed. And we will discuss more technical details regarding this particular system in the next couple of slides. Traditionally, people will conduct single B-cell sequencing to recover the antibody variable region sequence after recovering the positive B-cell by the single B-cell screening approach, followed by recombinant antibody production to generate purified material for binding and function assays. However, this workflow will consume a significant amount of resources and time, especially when you have to deal with hundreds, even thousands of single B-cell clones. Instead, we adopted a technology called ProSpeed Linear Expression Cassette, by which we only need to do PCR to clone the antibody variable region into a linear vector and go straight to transient expression without any needs for sequencing, gene synthesis, molecular cloning, and plasmid purification. The transient transfected supernatant will be screened by function assays to narrow down the candidates in a much quicker way. The overall timeline with this optimized process only takes about 2 to 3 weeks, comparing to 5 to 6 weeks with the traditional workflow. Here is an example of using the ProSpeed system to identify the functional antibody candidates against interleukin-8. The isolated B cells were first screened by on-chip binding assay against both human and cyno-monkey protein antigens. A total of 116 binder B cells were exported for high throughput expression without sequencing. 85 of them were confirmed as double high binders by ELISA, followed by the cell-based RGA assays. Finally, 16 candidates with receptor ligand signaling blocking capabilities were recovered. Next, I will share a case study to directly compare the screening efficiency between phage display library versus single B-cell approach. After alpaca immunization, the PBMC from the immunized animals is split into two halves with equal number of cells. One of them is for phage library and the other for single B-cell screening. We identified 68 double positive clones against protein and cell line antigen from the phage display approach, and finally end up with 36 unique sequences confirmed by both ELISA and flow cytometry using the recombinant purified antibody. In parallel, 460 positive B cells were identified as double positive against protein and cell line antigens. 164 of them, which is only 1/3 of all positive B cells, were selected for recombinant expression by the ProSpeed linear expression cassette. Then 92 clones were considered as high binders for both human and cyno antigen cell line. And finally, 62 unique sequences were recovered. Overall speaking, the hit rate of the single B-cell approach is roughly 5-fold higher than the phage display approach. Besides the general performance, we also looked into the sequence diversity of candidates from both platforms. The green bar represents the single B-cell approach, while the brown bar stands for phage display library. Although the distribution of a V gene germline is identical in both platforms, the dominant germline, which takes up roughly 40% to 50% of all candidates, is different from one to another. In addition, based on the analysis for distribution of CDR3 lengths on the right panel, we also noticed that even with both platforms generated candidates with varying CDR3 length, single B cell approach tends to recover sequences with longer CDR3 with the length beyond 22 amino acids, indicating a better paratope diversity. We further looked into sequence alignment of all candidates from these two platforms. Phage as blue and the single B cell as green. Based on the phylogenetic tree analysis, there is only one overlapping hit from these two sets of sequence. Furthermore, although some antibody families are shared by both platforms, each strategy also contributes to exclusive families. Under such circumstances, combining two different discovery approaches will certainly improve the chance of finding novel candidates with desirable profile. Finally, we compared the affinity by Beercore SPR kinetic analysis. The affinity KD data of leads from both platforms showed picomolar range, with top 5 candidates demonstrating similar affinity to the IgG clinical benchmark. Next, let's move on to the second part of this case study, a proof of concept study of a T-cell engager prodrug targeting the ENPP3. T-cell engager has been widely used for hematological tumors. However, its efficacy in solid tumor is still limited due to the lack of tumor-specific targets, leading to an increased risk of on-target off-tumor toxicity, the OTOT. One of the approaches to address this challenge is to build a prodrug T-cell engager that is conditionally activated in the tumor microenvironment. The prodrug T-cell engager comes with a masking domain that blocks the interaction between target and the antibody. The mask domain is fused onto the T-cell engager by a protease-sensitive linker. Upon entering the tumor microenvironment, the enriched protease will digest the cleavable linker, leading to the tissue-specific release of active form of T-cell engage. This strategy will create a wider therapeutic window to achieve a balance between the effective dose and unacceptable toxicities. Using the antibody discovery strategy as described in the previous session, we identified a panel of a VHH antibody targeting CD3 and ENPP3 as the building blocks for the T-cell engager. The affinity of CD3 antibodies as evaluated by flow cytometry on the left panel and the Buccal SPR on the right panel showed a diversified profile comparing to the clinical benchmark SB34. In addition to human CD3, most of these candidates also demonstrated decent cross-reactivity against the cyno-monkey CD3 as evaluated by ELISA against the recombinant protein and flow cytometries against cyno-monkey pan T cells. Similarly, we also evaluated antigen binding affinity of top ENPP3 candidates by flow cytometry against human and cyno-monkey cell line, and also the KU812 cancer cell line. These flow cytometry data strongly suggest better binding affinity of the candidates comparing to clinical benchmark XmAb 819 from Xencor. After identification of a VHH antibody against both arms, the CD3 and the ENPP3, we moved on to the discovery of a masking peptide. Among a panel of peptide candidates, two of them, the peptide A and B, were selected for its capability to block the interaction between CD3 protein and CD3 VHH antibody. Later on, these two peptides were fused onto the CD3 VHH and validated by both ELISA and T-cell activation RGA assays. In both assays, the peptide A demonstrated potent inhibition of a CD3 VHH antibody binding to CD3 protein as measured by ELISA, leading to the total silence of a CD3-mediated T-cell activation as measured by RGA reporter assay. After characterization with each of these three building blocks, we constructed the bispecific T-cell engager prodrug candidates using a bivalent symmetrical format as indicated on the lower left panel. The masking peptide is fused onto the N-terminus of a CD3 VHH, followed by ENPP3 VHH and the human IgG1 FC. This candidate was first validated for its protease-induced activation in both CD3 ELISA binding assays and the T-cell activation RGA assays. The prodrug showed significantly reduced activity comparing to the XmAb 819 and the unmasked active T-cell engage. Upon protease treatment by MTSP1, MMP7, and MMP14, the activity was greatly restored. And the MMPs is much more potent than the MTSP1 because of the design of the cleavable linker. Later, we observed similar trending of activity in the TDCC assays. In this assay, the Jagger cell was replaced by PBMC as the effector, while the target cell is still the KU812 cancer cell line. Again, the prodrug demonstrated inducible activation of a T cell upon protease treatment, while the enzymatic activity is ranked by MMPs followed by MTSP1. Since we are using a bivalent symmetrical format for the T-cell engager, the spontaneous T-cell activation in the absence of a tumor-associated antigen is certainly a major potential risk. In this assay, we are monitoring the undesirable activity by the expression of a T-cell activation marker CD25 and CD69 on the CD4 positive and the CD8 positive T cell using two independent donor samples. In all 8 panels, the prodrug candidates showed minimal T cell spontaneous activation comparing to the bivalent SP34 and the unmasked active T cell engager. Besides functional activity, we also evaluated the antibody polyreactivities by BVP ELISA and AC-Sins. In both panels, the prodrug candidate demonstrated reduced score as indicated by the brown bar comparing to the system control, the XmAb 819 and the unmasked T-cell engager, suggesting a lower risk of polyreactivity. After in vitro profiling, we also looked into the inducible activation by ex vivo system. In this assay, the prodrug with non-cleavable linker on the left panel and the cleavable linker on the right panel were pre-treated with conditioned media from the culture of a patient cancer tissues, followed by the measurement of T-cell activation using the RGA report assays. Comparing to the non-cleavable version, the prodrug with cleavable linker exhibited diversified activation profile upon incubation with conditioned media as labeled with these different code names. This data further demonstrated the capability of a conditional activation by prodrug. Finally, let's move on to the in vivo efficacy study. In this CDX model with a human PBMC transplant and the KU812 cancer cell line, we set up an escalating dosage from 1x to 100x for the prodrug. Comparing to clinical benchmark XmAb 819, the prodrug with 100x dose showed significantly better tumor growth inhibition, the TGI, while the systemic toxicities is well under control by the measurement of a body weight. In addition, prodrug with non-cleavable linker showed minimal TGI similar to the vehicle control. This data further confirmed the feature of inducible activation by the prodrug. Later, we further looked into the detailed profile of toxicities by non-human primate monkey studies. In this study, the prodrug was dosed at 10x and 100x respectively, comparing to XmAb 819 and unmasked active T-cell engager at 1x dose. In the PK profile, the active form of a prodrug, as indicated by the red dashed line and for the 10x dose and the green dashed line for the 100x dose, was gradually and sustainably released with a greatly reduced peak concentration and delayed exposure, suggesting a reduction of systemic toxicities. Further analytical data actually confirms our theory in previous page. This is a high-level summary for clinical observation of these monkeys, and those red colors highlights all abnormalities. Comparing to the XmAb 819 analog, the prodrug with 100x dose showed the least amount of adverse effects, including vomiting, upregulation of hematology indicators, urine analysis indicators, and ADA positive rate. Cytokine profile further demonstrated the safety profile of the prodrug candidates in these studies. Although all monkeys showed a transient increase of multiple cytokines at 4 hours after dosing, they all recovered to pre-dosing levels by day 2 to day 8. What's more striking is the cytokine induction by prodrug, as indicated by the color of red and green, was much lower than XmAb 819 and the unmasked T-cell engager during the whole period. With all of the data above, we can confidently conclude that the prodrug offers a combination of a better efficacy and lower toxicity profile. Before I end this discussion, I would like to say a few words about ourselves. The discovery group is one of the very first team established under the wing of GenScript. Over the last 2 decades, we have delivered more than 2,400 projects in multiple indications, including oncology, immunology, and metabolism diseases. With our target to PCC one-stop solution by multiple platforms, we're hoping that we can become your reliable business partners with speed, quality, and cost efficiency. Okay, that will be all for my first talk. Let's move on to the Q&A sessions.